Thanks everybody for joining us today. So today I'll be telling you a bit about our recent work developing combination treatments for Chagas disease and how we think they can be really quite transformational for patients. So the work I'll be presenting is work carried out by a large group of people. So this work carried out by University of Dundee, very close collaboration with GSK, and also including the NDI and the London School for Hygiene and Tropical Medicine. So I'm going to start with a very quick introduction to the drug discovery unit. So let me get the marker up. So the drug discovery unit at University of Dundee is quite unique in that it is a little bit like a small pharmaceutical company embedded within a university. So we have all the infrastructure and the skills and expertise to develop drugs, but we can do this in a non-profit situation in a university. And that takes us really to our mission, which is to develop medicines for unmet medical need, but in particular for tropical infectious diseases. So we work on a range of diseases, and they're listed on the left there. So you can see that's mainly neglected tropical diseases, definitely all tropical diseases with big impact in low and middle income countries. And at the early stages of the process, we tend to work with academic collaborators who understand the biology of the pathogens very well. And then we apply our drug discovery expertise to these projects to ultimately deliver what we call clinical candidates, so compounds that are ready to go into phase 1 clinical trials. As I already mentioned, the project here is a close collaboration with GSK. So we've been working with GSK since 2011 in Kinetoplastid Drug Discovery as a single team, really. And it's also closely we also work closely with the NDI in this space for this project. So in terms of impact that we've had in tropical diseases, so we've got currently 4 compounds in clinical trials. 1 in phase 2 for malaria, 2 for visceral leishmaniasis in phase 1. And then there is a cancer treatment at the bottom, which seems a bit out of place, but actually this cancer treatment came from an African sleeping sickness program that we were developing, and these compounds are now also moving to phase 2, so that's very exciting. So let's talk about Chagas disease. So very quick introduction. So Chagas disease is caused by Trypanosoma cruzi. It's an intracellular parasite, so it lives inside the cytoplasm of the host cells. And it has a very wide tissue tropism, so it can essentially infect any cell in the body of a patient. The parasites are spread by these triatomine bugs. And so this disease affects millions of people, mainly in Latin America, with approximately 7 million people infected. And the number of deaths is pretty well underestimated, is not well known, but some of the estimates are that over 1 million people, for example, have heart disease caused by this parasite, which is very significant. So key point to make when it comes to Chagas disease is that this is a chronic disease. So what you can see on this plot here is that after an initial burst of number of parasites, the immune system in an infected person can control the parasites quite well. But what happens is that a small number of parasites persist in the person, and that very small number of parasites eventually 10, 20, 30 years later causes the disease, which mainly manifests itself as heart disease, but can also show mega organ disease. So what is important from a drug discovery point of view is that we have a very small number of parasites causing the disease. And so what we think we need to do to develop a good new treatment is have a treatment that can kill every single parasite. Because if every parasite is gone, then of course symptoms should not arise. Together with that, it's important to treat early in the disease, because if you treat when people have symptoms, even if you kill the parasites, it's very difficult to reverse the symptoms. Good. So there are treatments available, benznidazole and nifurtimox, but they have some significant issues, mainly the long treatment duration that's required, as well as the side effects, which essentially prevent people from taking the treatment or stop people taking the treatment if they have these effects. So I'll go straight into the program. So this is a drug discovery program focused on one particular series of compounds, which we call the 220 series. So you can see a couple of examples shown here. So I'm not going to go into the details of the whole chemistry program. I'm going to talk mainly about the biology and the efficacy of these compounds. But you can see there is a pyrazole pyrimidine core on these compounds. that's shared between all the compounds, and then we have these different groups here on the top right. So if we look at this biological profile shown here in the table, you can see that these compounds are potent against intracellular hematogogues. So these are PEC50s, but so the higher the number, the more potent. 8 means that this is an EC50 or an IC50 of 10 nanomolar. So active against the intracellular parasites, inactive against the host cells. So, that's good. We don't want to kill the mammalian cells. Active against non-dividing trypomastigotes, so another form of the parasite that we want to kill. And importantly, these compounds are inactive against CYP51. So CYP51 is a target, or is the target of posaconazole and fosravuconazole, 2 compounds that failed in the clinic, and so we try to avoid progressing compounds with that target. So we were very keen when we saw this biological profile in our series, and we wanted to progress these to animal efficacy studies. So the first step we take is to study the pharmacokinetics of these compounds. So essentially, we dose compounds orally to mice, and we then see how much compound we get into the blood. But first, I should maybe point out here the properties of this series. So these are just standard properties we measure. So lipophilicity is okay, solubility is pretty decent, and metabolic stability is all pretty decent. So overall, quite a good profile, a promising profile to take forward to animals. So on the right, you can see the outcome of a pharmacokinetics experiment with this compound shown here, where we dosed mice orally with either 25 milligrams or 50 milligrams per kilogram. And so all the data shown in blue and red here are the free levels, so the non-protein bound levels of compound. And that's important because it's the non-protein bound levels of compound that can reach the parasites. So these are levels in blood, and what you can see is the levels are high with respect to this dotted line here, which is the EC90 or IC90 from our cell-based assay. So what this is telling us is that we have a much more compound in the blood than we need essentially to kill the parasites. So that's a good sign. That tells us that we should see efficacy if these compounds work. So based on this pharmacokinetic data, we progressed the compounds to an animal model of chronic Chagas disease. So if you focus on the top right here, that is a little outline of the model. So we infect mice. They go through the acute phase and then enter the chronic phase. And so around 100 days after infection, we start dosing with our compounds. So in this example, we're dosing for 21 days. And at the end of dosing, we go through 3 rounds of immunosuppression. And so remember, we have a very low level of parasites. And so what we try to do with the immunosuppression is give the parasites a chance to start growing again and make it easier for us to detect the parasites. This is a bioluminescent model, so we can actually see light from the parasites coming out of the living animals. And so what you see on the left here, all this color, that is the signal coming from the parasites in the animals. And so this is the acute phase. And then, with day 93, day 99 here, we start the treatment. And you can see at the end of the treatment, we basically don't detect any signal anymore. So because of the drug, the parasites are not visible anymore. However, once we go through the rounds of immunosuppression, you can see that at the end of the experiment, we are starting to see signal again. And so what we observed for this series, shown here for compounds 2 and 3, but for many more compounds, is that while we could prevent relapse in some of the animals, there were always animals relapsing. Now, remember, what we are looking for is a compound that can achieve sterile cure, where there are no relapses in any of the animals. And so this was obviously a disappointing result and suggested that with this series, we could not achieve the cures that we were looking for. So we tried to understand why that was, did a series of experiments, for example, looking at cytality. So here we look at a rate of kill experiment in an in vitro culture. And what you can see is that these slopes are negative, meaning that the number of parasites is indeed declining. So these compounds are cidal, which is good. So yeah, I was just showing here that we had compound 3, which could prevent relapses. And so the key question at this point was what the target might be of the series, in particular to understand what human toxicities there might be seen. So a lot of work summarized in this slide here, carried out by Susan Wiley in the mode of action group at the University of Dundee. And basically, this work showed that complex 3 or cytochrome BC1 in the electron transport chain in the mitochondria was the target of these compounds. So this is, of course, important for ATP generation. You need to build your proton gradient. And so through inhibition of this, we were killing the parasites. Now, humans also have complex 3. And so it was critical for the rest of the program to develop compounds that were highly selective for the parasite cytochrome B. So what you see here is compound 3, which I've shown you before. And so compound 3 does inhibit the human complex 3. And so a lot of medicinal chemistry is summarized in one slide, going from this compound to the compound on the right, number 4, which was completely clean and very selective against the human mitochondria. So at this point, of course, we had to check that this compound was still efficacious. We tested it at multiple doses, 50, 30, and 10 mg per kilogram. And what you can see here is ultimately that these mice were all clean, no relapses. You can see here on the plot as well. So no relapses in the combination arms, whereas nearly all relapses in the monotherapy arms. So this is really where I'll stop. So we think this is potentially transformative for patients. We think we could, with this combination therapy, reduce or avoid the adverse effects. And we also think we can reduce the treatment duration. A compound from this series is currently being progressed by the NBI towards phase 1 trials. So that hopefully will give us the tools to test this in the future in people. So I just went quickly, but I think hopefully that's the key messages conveyed. So this work was recently published in Science Translational Medicine, so you can see all the people involved on this slide. So I'll thank you for your attention, and hopefully we have time at the end of the session for some questions.